Hey there, everybody. This is the first half of Mendel and the gene idea. So it's going to be talking about Gregor Mendel. This is chapter 14, which is all about your Punnett squares, different forms of inheritance. This is probably one of the simpler chapters that we're going to be going over. So we're splitting it up, half of it today and then half of it tomorrow. So if you have any questions, please come in a class tomorrow and I'll be happy to answer them. But we're going to go roll right into it. So this is just a little bit of background information about Gregor Mendel. He was an Australian monk, and he was interested in genetics, and he specifically looked at pea plants in order to study inheritance. The reason why was that he was able to control mating by having self-fertilization versus cross-fertilization taking place. So in self-fertilization, pea plants, because they're hermaphrodites, they have both the sperm and ovaries, or sperm and eggs, in order to produce offspring. To prevent the pollen from reproducing with the plants, he would end up snipping off parts of the plant that would actually produce the pollen. So that way only the stamen would survive in order to control what pollen would actually go in there. So he made sure that self-fertilization did not end up taking place. And then from there, he would bring the pollen from one to the stamen to the other, so that way he would be able to actually have the controls take place. So he actually got to control what got mated with what. And there are many different varieties that were available, different traits that he ended up taking a look at. Also, the generation time for pea plants was relatively short. Think like our Wisconsin fast plants. I mean, it would be done relatively soon, within like a few weeks. So a couple of different experiments that he did was his experience with true breeding plants. So he had purple flowers and white flowers. True breeding meant that if you cross a purple with a purple, you get purple, and a white with a white would get white. Well, he decided to cross a purple and a white flower, and when he ended up checking it out, he saw that they all had purple flowers. So it's like, okay, well, let's go ahead and see what happens if we were to self-pollinate these or cross-pollinate them. So he would cross these purples hybrids because they're purple and whites, and the hybrids would end up showing purples as well as, excuse me, it would end up showing white flowers as well, which kind of baffled him because it's like, well, wait a second, the white disappeared from P generation to F1 and then showed back up in F2. Why the heck did the white flowers pop back up? So originally we used to believe in this thing called the blending hypothesis, which was that you're always a blend of whatever the traits were. And I mean, this is what we thought about with parents and kids, right? Because you kind of look like a combination of your mom and your dad, and plenty of other things work like that too. Well, sure enough, Gregor Mendel was able to reject, or we were able to have the idea that we rejected the blending hypothesis and instead had this thing called the particulate hypothesis, which is a very fancy way of saying that you're passing on factors that end up determining traits, i.e. we're passing on genes that end up making these traits. So, but this is what he ended up finding. Like, this was his experiment that really led him to kind of understand this idea of all these laws he started to come up with. So a term you need to be familiar with is an allele. So alleles are alternate forms of a gene. So the idea here is that there was an allele for purple flowers, and on the same locus, the same location on the chromosome was an allele for white flowers. So in the case here, this plant had two purple alleles, this plant had two white alleles. And then when we talk about this heterozygous and the P1 generation, had a purple and a white. So these are just different characteristics that he ended up taking a look at, and he found that some of them were dominant, the other ones were recessive. So the ones that are dominant are located here on the left side, the ones on the right are the recessive versions of these traits. Mendel's principles, so he found that there were alternate versions of genes, which are called alleles, which cause variations among inherited characteristics, whether you got the dominant or recessive. For every character, each organism inherits one allele from each parent. So you get one from from one of them, you get one from the other, and then of the two alleles, one could be dominant, the other one could be recessive. If we go back to this picture right here, the idea is that there are two dominant alleles here, 2 recessive alleles here. You're getting 1 from each of your parents, but then the dominant one is superseding the recessive one, which is when we're talking about the law of dominance. And the law of segregation means that during gamete formation, the alleles get separated. Now, if you kind of think back to this in meiosis, what happens is that our homologous chromosomes, which are the chromosomes with the same alleles, they have the same genes on them, they get segregated into 2 different haploid cells, and then the sister chromatids are then separated in half from there. So that's the law of segregation. Actually, that's how we're able to track it, is during meiosis 1. But yeah, the 2 alleles, they end up getting separated during gamete formation. So this is what the law of formation segregation ends up looking like, is that you get this 2 p's here, 2 little p's there. You're only getting 1 of them. This purple flower had 1 or the other. And then from there, when we cross these guys, there's a dominant and a recessive, a dominant and recessive. They get segregated, so the sides on this get the capital P from up here. So this capital P goes all the way down there. This little p goes all the way down there. It'll be a little bit easier to see in this next image that we have here, or coming up. So we have dominant traits or recessive traits. There's also other terms to be familiar with this: homozygous and heterozygous. Homozygous means that you have 2 of the same alleles, so they can be 2 dominant alleles or 2 recessive alleles. Heterozygous means that they are different, so you have a dominant and a recessive. So homo meaning same, hetero meaning different. So we also have the terms phenotypes and genotypes. Your phenotype is what we physically see, while the genotype is going to be what the genes actually are coding for. So in the case here, we can see that we have 3 different genotypes. We have capital P, homozygous dominant. Here we have heterozygous, and then we have homozygous recessive located right here. But these different genotypes, some of them can code for the same phenotype. So in the case here, this homozygous dominant and heterozygous both will code for purple right here, while the homozygous recessive is only going to be coding for the white right there. You will be asked to find like genotypic ratios or phenotypic ratios. Genotypic ratios is talking about how many homozygous dominant you have. You have 1, which is right there. How many heterozygous? You have 2. It's located right there, and recessive right there. Phenotypic ratio is saying how many purples. You got 1, 2, 3, 3 to 1 white right there. So they're just ratios of being able to describe your findings. This is a better idea of showing you the law of segregation, but what we're talking about with Mendel. So here are the alleles for the parents. So again, keep in mind, these would be homologous chromosomes right there. They get separated. So here is the first cell over here, then here's the second cell. This is the first split after meiosis 1, and then we'll just say that there's another split, but you have 1 allele, so we'll say the dominant allele on the first homologous chromosome, and then you have the recessive allele on the second homologous chromosome. And this is why meiosis and genetics are so tightly wound together. What happens is that when you set up your Punnett square, you're going to be putting your, they're going to be traveling down. So it's actually color coded for you to be able to see. So the purple P's starting from up here will be dragged down to these 2 boxes. The orange P's are going to be dragged down to these 2 boxes. This brown P is going to be dragged over to these boxes, and these blue P's are going to be located over to here. So you can see the different ratios are located right there. You can use a test cross in order to help identify an unknown individual. So the way that this would work is that I have a purple plant right here. I don't know if it is homozygous dominant or heterozygous, but what I can end up doing is crossing it with a homozygous recessive. And only 2 results will take place. If I have a homozygous dominant and a homozygous recessive, the outcome would be all purple if it's homozygous dominant. So this would be able to tell me like, hey, this unknown plant was homozygous dominant. Versus if this one was heterozygous, if it was heterozygous with a homozygous recessive, the test cross would show that half of them would be purple and the other half would be white. This is just our way of being able to identify unknown genotypes. Law of independent assortment is saying that each pair of alleles gets separated individually during gamete formation, so color is separate from shape. In the case here, we can see that we have a yellow round P, which is capital Y, capital Y, capital R, capital R, and we have a green wrinkly P, which is little y, little y, little r, little r. We end up creating some gametes right here, and then we have this F1 generation, and then we'll go ahead and do the self-pollination, and then we'll find that the sperm and the eggs will give a lot of very different variations of yellow round, yellow green, yellow wrinkly, and yellow green wrinkly. And then we can talk about the results, our approximate phenotypic ratio. So we'll give you an opportunity to practice some of these tomorrow after we work on the project a little bit. I'll also attach a video on how to solve these two-factor crosses. But, sorry. You have monohybrid crosses and then dihybrid crosses. The difference being is how many characters are being tested. So here would be a monohybrid cross. This would be a dihybrid cross, meaning that one-factor or two-factor crosses. And this is what a dihybrid cross looks like. This is when we end up using our foil to get all of our different gamete combinations, and then we have our Punnett square get filled in like so. So capital R, capital R, capital R, capital R, Y, 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 Y right there. Here we have two big R's and little y, little y. Here you have big R, big Y, little y right there. So this is just our way of being able to read and figure out what our actual outcomes are going to be. So if you have any questions, please come in tomorrow and I'll be happy to answer them for you. Try out the practice problems. Make sure you can do a basic Punnett square as well as a two-factor cross. And if you need to have review on how to do a two-factor cross, it would be perfectly helpful or be perfectly fine to go over that in class tomorrow. Thanks for watching this, and I'll see you guys tomorrow. Bye.